Hello, I'm Meredith Blackwell, and this is Paola Bonfante, and we're at the International Mycological Society, or International Mycological Congress 11 in San Juan, Puerto Rico. And it's very nice to have Paola to interview because she was our keynote speaker for the whole Congress. Would you like to tell us something about the tradition of mycology in Italy? And we certainly want to hear about your own work too.
Yeah, thank you. Thanks for the opportunity. It is really very nice to remember that Italy has really a great tradition in mycology, starting from the very past times where we can remember, for example, the famous name of Saccardo, since a lot of identification of microfungi are really coming from his studies. But in Torino we had a long tradition of interesting mycorrhizal fungi, and for that I would like to remember that usually we think that the first person who described ectomycorrhizas is the famous Professor Frank in Germany.
Yes.
But it is quite nice to remember that exactly in the same years, Gibelli, professor of plant pathology in Bologna, did very beautiful drawings showing the ectomycorrhizas in oak roots, and therefore starting from these first observations, many groups developed this type of research. For example, in Torino there was a strong interest in truffles as hypogeous fungi, and Professor Mattirolo, who was the director of the botanical garden at the start of the 19th century.
There's a termite fungus that I've worked on that's named for him.
Yes.
Yep, termite ectoparasite.
Yes,
Sorry to interrupt.
very nice, very interesting. So he was the first to propose that maybe also truffles were ectomycorrhizal fungi. And it is quite nice since we have a sort of painting, and I am used to say that this is a sort of a first poster of a scientific history of maybe mycology in Torino. Since in Torino there was an international exhibition, which was a very common habit in the start of the past century, and so there was also interest for science, and they prepared this painting where we can see the people looking for truffles associated and the mycorrhizas. So it's a very nice document. But after that, in Torino, there was a strong group interested in microfungi. And so I would like to surely mention the name of Beniamino Peronel, since he already around 1920, for example, developed a sort of soil assay plate. I mean, to collect the soil, to dilute, and to grow the fungi, to isolate the fungi growing from the soil. And there was really strong groups working on this field, and I would like to remember some people like, for example, Anna Maria Lupi, Anna Fontana. They, for example, isolated many saprotrophic fungi from soil, and I was very amused during this meeting, since, for example, they used the dung of rabbit cows as an important source for studying nitrifolous fungi. And on this basis, in 1950, they funded, put the basis for a huge fungal collection. And so they funded what is called the Mycoteca, the Mycotech. And at the moment, this collection of fungi is considered one of the most important in Italy. We have a huge number of isolates, and the current researcher who is following this is Cristina Varese. She is in this meeting. And they did really a great work. Since at this moment all the taxa have been characterized also from a molecular point of view, so it is really quite important collection. And Cristina Varese has recently really coordinated some European projects which are really focused on the importance of this fungal collection. So this is one part.
Yeah.
Well, by contrast, on the other hand, in Italy, of course, we have a strong interest in mycorrhizal fungi, ectomycorrhizas, of course, mostly truffles, since you know that in our country we are more or less we eat fungi.
Okay.
We eat, I do not know.
Mycophage.
Exactly. Exactly, and so truffles represent a big business since they are very, very expensive. And following this direction and following really tradition of our department, we have continued this type of studies with many colleagues now working in the lab. But for example, we were also involved in the genome sequence of truffles together with Francis Martin. By contrast, I was mostly interested and mostly focused my attention to mycorrhizal fungi, and this following my first mentor, Anna Fontana, and then Silvano Scannerini. And on this line, I developed different approaches, starting from cell biology, which was my first approach. Then I developed molecular tools, and now we are doing genome sequencing. And so I would like to say that all our group, both belonging to university, both belonging to National Council of Research, is mostly working on mycorrhizal fungi. And really we also try, thanks to some applicative projects, to move our research from the lab to the field.
Oh, a lot of people do just the opposite today.
Yes. And so this is maybe the main point.
Yeah. Well, that's really interesting. We don't, I guess in our mycology courses here, we don't emphasize the Italian work so much, except for Sicardo, but it certainly has been very important.
Yes.
But also, the other thing that we know is all of the wine yeast. That's been an important industry too.
This is true. And indeed, surely there is another important collection in Perugia, which is in the center of Italy, where they have a very, very good collection of yeast. And of course, this has also a lot of relevance also from the industrial application, biotechnology. So this is, of course, another important point.
Well, you have a wonderful ongoing career, and I have heard you speak at Asilomar at the fungal genetics meeting.
Yes.
I think I was in a symposium with you, but you outdid us all, and you were kind enough to give us your tape of a fungus going through a plant.
Yes.
It was wonderful.
Thank you.
And then the second time I heard you was at the European fungal genetics meeting in Paris. Yes. And then now again. And so here, 3 times I've heard you speak, and never on the same topic, and all very interesting.
Thank you.
We all enjoyed your little run through all the IMCs and your little video. It was a really nice presentation. And I appreciate so much your taking the time today to talk to us.
No, thank
you.
you.
As our keynote speaker.
No, thank you to you. It was really great pleasure. Thanks.
Yes.